I want you to imagine your brain for a second. Picture it not just as, a biological organ, but as this bustling, perfectly organized city.
Right, complete with infrastructure and traffic.
Exactly. You have millions of commuters, these are your neurons, firing off messages, racing down the highways. You have delivery trucks dropping off nutrients. And crucially, you have this massive, highly efficient sanitation department keeping the streets completely spotless.
Which is vital because brain cells generate a lot of waste.
They really do. But in Alzheimer's disease, it's like there is a sudden, catastrophic sanitation strike, a slow, insidious buildup of toxic cellular trash just starts piling up in the streets.
Specifically, we're talking about sticky amyloid-β plaques and tangled tau proteins.
Yeah, those are the main culprits. And over time, the roads get completely blocked, the power grid flickers, and eventually whole neighborhoods just start shutting down in the dark.
And it's a devastating reality. I mean, it's why Alzheimer's is the 6th leading cause of death in the U.S. alone.
It's terrifying.
It is. The frustration for patients, their families, and frankly, the whole scientific community is that our current treatments are mostly just offering temporary symptom relief.
Right, or marginally slowing things down.
Exactly. Or they act like single-target snipers. We have drugs that can clear out one specific type of trash, like the amyloid plaques. But if your city is collapsing from multiple infrastructure failures, you don't just need a bigger dump truck.
Sweeping the streets once doesn't fix the underlying broken systems.
Precisely.
Which is exactly why today's deep dive is so incredibly exciting. We're looking at a fascinating new research paper detailing the discovery of a small molecule. Right now, it's going by the very catchy scientific name of DDL-357.
Catchy indeed.
Rolls right off the tongue. But the sheer scale of what this molecule might do is honestly mind-blowing. Scientists essentially engineered this chemical not to attack the brain's trash directly, but to supercharge the brain's own natural garbage trucks and repair crews. Our mission today is to explore how researchers built this molecule from scratch and how it fundamentally upgrades a dying brain.
And I just have to say, the stakes with this kind of research are immense. Finding a multifunctional drug candidate, one that clears out toxic proteins while simultaneously, promoting neuroprotection and repairing the power grid, is the absolute holy grail of neurodegenerative research right now.
We're talking about a complete paradigm shift.
Absolutely. Moving away from just slowing the decline and moving toward a systemic reboot.
Okay, so let's unpack this. Before we can even talk about how this new drug, DDL-357, actually works, we have to understand the biological target it's trying to boost.
The foundation of it all.
Right. The researchers focused their entire effort on a specific protein in the brain called clusterin. But why start there? I mean, out of all the things going wrong in an Alzheimer's brain, why zero in on this one specific protein?
Because the genetics point us right to it. When scientists look at these massive genome-wide association studies for Alzheimer's, they find that a mutation in the clusterin gene is actually the 3rd strongest genetic risk factor for the late onset form of the disease.
Wait, the 3rd strongest overall? That's huge.
It is. And this specific mutation hinders the brain's ability to produce a specific secreted form of this protein, which we call sCLU.
Little s, capital CLU.
Exactly. And the clinical data is just crystal clear here. When your sCLU levels drop, you see a massive increase in those toxic plaques and tangles.
And the cognitive decline accelerates rapidly.
They got it.
So if we go back to our brain as a city analogy, this sCLU protein sounds like it's, the VIP bouncer and the head of the sanitation department rolled into one.
That's a pretty good way to look at it, yeah.
From what I read in the source, it physically binds to those toxic misfolded proteins and literally prevents them from clumping together into deadly roadblocks.
It's a fantastic chaperone protein, yes. But it does far more than just tackle the trash and hold it in place. It actively escorts the garbage out of the city.
Oh, wow. How does it do that?
Well, sCLU has a very high affinity for specific receptors on the borders of the brain. So it grabs a handful of toxic amyloid-β, drags it to the blood-brain barrier, and essentially hands it off to border control.
Wait, border control has a name, right?
Yes, specifically it hands it to receptors like LRP2.
LRP2, okay.
Right. So LRP2 helps transport it across the barrier to be thrown out into the body's periphery. But that's not the only way. Alternatively, sCLU can hand the trash over to the brain's own immune cells, the glial cells.
By binding to a different receptor?
Exactly, a receptor called TREM2. When it binds there, it triggers those immune cells to literally swallow and digest the toxic proteins in their lysosomes.
That is wild. So it's actively directing the cleanup crews, telling them exactly where to put the garbage.
Just a highly coordinated effort.
But the paper also mentioned it protects the neurons themselves, right? Like it's not just a garbage collector.
This is what's so fascinating about it. The sheer diversity of its mechanisms. sCLU acts kind of like a switchboard operator, too. It stimulates neurotransmission by interacting with a transporter called VGLUT1.
VGLUT1. So it's keeping the electrical communication lines wide open so neurons can successfully fire messages to one another.
Right. And furthermore, it triggers a cascade of chemical signals inside the cell, specifically through something called the renin signaling pathway.
The renin pathway, what does that do?
That pathway is crucial because it specifically blocks the harmful tangling of tau proteins.
Ah, the other type of cellular trash.
Exactly. Without this protective renin signaling, tau gets chemically altered. It's a process called phosphorylation.
Phosphorylation, right?
Yeah, and that's what causes it to form those cell-killing tangles. But sCLU stops that phosphorylation from happening. It even blocks the assembly of certain molecular complexes that would otherwise trigger the cell to just, well, commit suicide.
Apoptosis.
Right, exactly.
Okay, let's take a quick second to recap where we are, just to make sure we've got all these details straight.
Good idea.
So if you have low levels of this sCLU protein because of a genetic mutation, your brain is basically defenseless. The trash piles up because it can't get to the LRP2 or TREM2 receptors. The cells can't communicate because VGLUT1 is lacking, and neurons die because tau gets phosphorylated.
That is a perfect summary.
Which brings us to a massive biochemical puzzle. And I want you, the listener, to think about this. If your brain's natural defense system is completely underfunded like this, how do you give it a biochemical stimulus check? How do you force a dying brain to just make more of this specific protective protein?
That is the multi-million-dollar question right there.
You can't just guess what chemical might do that.
No, definitely not. So the researchers started with what's known as a high-throughput screening campaign.
High-throughput screening.
It's essentially a massive, highly automated biological treasure hunt.
I was wondering about this when I read it. How do you even test for that? Do you just start dropping random chemicals onto brain cells in a lab and hope for the best?
I mean, in a highly controlled way, yes. They took human brain cells. Specifically, they used a very resilient line of human glioblastoma cells called U87MG.
U87MG. Why those cells specifically?
They're a well-established cell line that naturally produces sCLU, so they're a perfect testing ground. They exposed these cells to thousands of different compounds from a massive chemical library.
Just looking for anything that worked.
Right. They were looking for any chemical that caused these U87MG cells to naturally secrete more sCLU. And out of thousands of attempts, they got 32 initial hits.
Only 32 chemicals out of thousands?
Yep. And through rigorous retesting, they narrowed those 32 hits down to 2 distinct classes of drugs. These are what we call epigenetic modulators.
Okay, epigenetic modulator.
One class was called HDAC inhibitors, and the other was called BA inhibitors.
Hold on, let me stop you there. Epigenetic modulators. Let me throw an analogy at you to make sure I am grasping how this works in plain English.
Let's hear it.
Okay, so if our DNA is the master instruction manual for building and running the city, these epigenetic molecules are like highlighters and whiteout, right?
That's a great way to put it.
Like, they don't change the actual printed words in the manual, they don't mutate your DNA, but they change which instructions the cell actually reads and executes.
I love that. Yes. To take it one step deeper mechanically, your DNA is incredibly long. Right, so it has to be wrapped tightly around proteins called histones.
Like thread around a wooden spool.
Exactly like that. When DNA is wound up tight, the cell can't read the instructions. Now, the body uses small chemical tags, specifically acetyl groups on lysine residues, to loosen that thread, exposing specific genes to be read.
Okay, acetyl groups on lysine residues. Got it.
Right. Now, Bt proteins, the things these chemicals are trying to inhibit, act as readers.
They read the tags.
Exactly. They scan the DNA, they recognize those acetyl groups, and they recruit a whole crew of transcription factors to turn on specific genes.
And I'm guessing the genes they turn on aren't always good.
Unfortunately, no. In the context of neurodegeneration, the genes the bed proteins turn on often promote severe, damaging neuroinflammation.
Ah. So by inhibiting these BET proteins with a BET inhibitor, you are basically hacking the cell's gene expression. You're rewiring it to stop the inflammation, and in the process, promoting plasticity and forcing it to read the instructions for making our protective sCLU protein.
You absolutely nailed it. That's epigenetics in a nutshell.
But wait, the researchers had a choice here. You mentioned they found HDAC inhibitors and BET inhibitors in their screening. Why did they ultimately go with the BET ones?
What's fascinating here is that the data made the choice incredibly clear. The BET inhibitors were vastly superior for a few critical reasons. First, they were incredibly potent.
Potent.
Well, in pharmacology, we talk about effective concentrations. The BET inhibitors worked at nanomolar concentrations. The HDAC inhibitors required micromolar concentrations.
Okay, put that into plain English for me. Nanomolar versus micromolar.
Think of it like watering a delicate houseplant. A nanomolar concentration is like using a precise eyedropper. It takes very little of the drug to get the exact response you want.
Okay, an eyedropper. And the micromolar.
The micromolar concentration is like needing a fire hose to water that same plant. You need thousands of times more drug to get the same effect.
Oh, wow. Yeah, that sounds destructive.
It is. As you can imagine, blasting cells with a fire hose of chemicals usually doesn't end well. The cytotoxicity test proved exactly that. The HDAC inhibitors were highly toxic to the cells at those high doses, so the BT inhibitors were crowned the winner.
Specifically, I think the paper mentioned a BET inhibitor called positive JQ1, right?
Yes, plus -JQ1. It had an incredibly potent response, an EC50 of 313 nanomolar.
Okay, so let's do another quick recap. We have a winner. We found a chemical eyedropper plus JQ1 that tweaks the epigenetic highlighters, calms down inflammation, and boosts our VIP bouncer, sCLU.
Exactly.
But, you listening to this right now might be thinking what I was thinking when I read this. You have a chemical that works beautifully in a plastic dish on some glioblastoma cells. But how do you get it into the heavily guarded fortress of a living human brain?
Oh, you've just identified the ultimate bottleneck in all of neurotherapeutics.
Here's where it gets really interesting. Because the brain has its own physical security system, right? The blood-brain barrier.
Yes, the BBB.
Yeah.
It is a microscopic wall of tightly packed cells lining every single blood vessel in your brain.
And it's tough to get past.
Evolutionarily, it's a masterpiece. It is designed to keep toxins, bacteria, and pathogens in your blood from ever touching your sensitive neural tissue.
But it's almost too good at its job.
It is so effective that it blocks over 98% of all small molecule drugs. Let that sink in. You could literally have the cure for Alzheimer's sitting in a syringe, but if it cannot physically slip past those tightly packed cells, it is completely useless.
That is so frustrating. So discovering the chemical was really just step one. Now they have to become like molecular locksmiths.
Precisely. They entered the drug design phase, the medicinal chemistry part of the journey. They took their best BT inhibitor, that plus DACU-JQ1 molecule, and used it as a foundational scaffold.
Like a chassis of a car.
Exactly. And over months of painstaking work, they synthesized 10 completely novel chemical variations of it.
How do you even do that?
They snapped different molecular attachments onto this core chassis, specifically using what we call ester and amide coupling reactions.
Ester and amide chemical bonds.
Right. They were essentially tweaking the molecule to see how changing its shape and properties affected its ability to sneak past the guards at the blood-brain barrier.
And then they run these 10 new creations through a chemical obstacle course, right? I think it was called the ADMET gauntlet.
The ADMET, it's a critical acronym in drug discovery. It stands for absorption, distribution, metabolism, excretion, and toxicity.
So basically, will it survive the human body?
Exactly. A drug has to be soluble so your body can absorb it. It has to survive your liver, which is constantly trying to break foreign chemicals down. We call that microsomal stability.
Makes sense.
And crucially, they have to measure something called plasma binding and brain tissue binding.
Which actually brings up a concept in the paper that totally shifted how I think about medicine, the free drug hypothesis. Explain why this is so critical, because it blew my mind.
It's a vital pharmacological principle. When you swallow a pill, a large percentage of that chemical acts almost like a magnet. It binds tightly to proteins in your blood plasma, or it crosses into the brain and just sticks randomly to the surrounding tissue.
It just gets stuck.
Right. And once a drug molecule is physically bound to something else, it is structurally locked. It is inactive. The free drug hypothesis dictates that it absolutely does not matter how much total drug gets into the brain. The only metric that actually matters is the concentration of the unbound, free drug floating around that is actually available to interact with your target receptors.
It's like Okay, it's like sending an army of 1,000 soldiers into the city, but 900 of them immediately get stuck in traffic or handcuffed to streetlights.
That's a great visual.
Only the 100 free soldiers can actually fight the battle.
Exactly. So the researchers needed a molecule that was potent, survived the liver shredders, slipped past the blood-brain barrier, and didn't get stuck to random proteins once inside.
A tall order.
Very. But out of the 10 candidates, one absolute champion emerged.
Enter DDL-357.
DDL-357. It was the only molecule to pass every single test in the gauntlet. It was incredibly potent. Its EC50 dropped down to 223 nanomolar.
Even better than the original.
Right. And when they tested it in living mice, it showed incredible blood-brain barrier permeability. But most importantly, applying that free drug hypothesis we just talked about, the unbound active concentration of DDL-357 floating freely in the brain was measured at 457 nanomolar.
Which is well above the threshold needed to actually trigger the brain to make more sCLU.
Way above it. It was a massive success.
Okay, let's pause and recap again. You, the listener, now have a highly stable chemical fortress crasher that safely reaches the brain.
Right.
But fixing the biochemistry on a cellular level doesn't automatically mean the lights come back on for the patient, does it? Like, did this highly engineered molecule actually fix the memory problems associated with Alzheimer's?
That's the real test. To answer that, you have to move from chemistry labs to complex living systems. So the researchers initiated trials using mice that were genetically engineered to develop Alzheimer's symptoms.
And they didn't just use one type of mouse, right? They ran 2 distinct models to be incredibly thorough.
Exactly. Because remember, Alzheimer's has 2 main types of trash: amyloid plaques and tau tangles.
Right.
First, they ran a 2-week subchronic study on a specific mouse model called ApoE4TR5XFAD.
Catchy names everywhere today.
I know, right? These mice are engineered to produce massive amounts of those toxic amyloid plaques. The goal here was just to prove the chemical mechanism worked in a living, breathing creature.
Did it?
It did. They gave the mice DDL-357 orally, and after 2 weeks, analyzed the brain tissue. They found a statistically significant increase in our protective protein, sCLU, right in the hippocampus.
The hippocampus, which is the brain's main memory center.
Exactly.
Okay, so the drug works inside a living brain just as it did in the dish. But like we said, Alzheimer's isn't just amyloid plaques. It is also those toxic tau tangles killing the neurons from the inside.
Right, so for their next test, a longer 6-week chronic study, they used a different mouse model called 3XTGAD.
And these ones mimic the tau tangles.
Yes. They developed both plaques and tangles. And after 6 weeks of oral doses, the results were incredibly promising. The mice showed a massive reduction in toxic tau phosphorylation.
Specifically at a site called SIR396, if I remember the paper correctly.
Spot on. SIR396. The drug was actively stopping the tangles from forming at that exact site. They even found a direct correlation. As the protective sCLU levels went up, the toxic tau went down.
Okay, the spreadsheets look great. The biochemistry is flawless. But I have to push back a little and ask the human question here.
Okay, go for it.
If we clear this microscopic cellular trash, does the mouse actually feel or think better? Like, can it remember things it couldn't before?
And this is where behavioral testing is so crucial. They used a specific protocol called the Barnes maze.
The Barnes maze.
Yeah, it tests spatial learning and memory. Imagine a large, brightly lit circular table with 20 holes around the edges.
Okay, picturing it.
But only one hole leads to a safe, dark box underneath. Now, mice hate bright, open spaces. Their natural instinct is to hide. To find the safe escape hole, they have to look at visual cues placed on the walls of the room, like a big square or a triangle, and use those landmarks to memorize the location of the escape hatch.
It's basically like trying to remember where you parked your car in a massive mall parking lot by looking at the light pole numbers. It requires complex spatial mapping.
That's a perfect analogy.
So how did our treated mice do in the parking lot?
The mice treated with DDL-357 performed significantly better than the untreated Alzheimer's mice. During the training phase, they learned the location of the safe hole much faster.
Wow.
And more importantly, during the 48-hour probe phase, which tests long-term memory retention, the treated mice found the safe hole much quicker.
So they retain the information.
Exactly. Their spatial memory wasn't just slightly better on a biochemical chart. It was tangibly, functionally improved in the real world.
They remembered where they parked their car. They literally got their memory back.
They did.
But how? I mean, we know the drug cleared the trash, But the researchers wanted to know exactly what the drug fixed under the hood to restore that kind of complex memory.
They did, and to figure that out, they ran what's called a proteomics analysis.
Proteomics?
Yeah, they basically popped the hood of the mouse brain to catalog every single protein that was altered by the drug.
And the discoveries here are what elevate DDL357 from a simple trash collector to a systemic brain upgrade, right?
Absolutely. This goes back to what we were talking about at the very beginning of the deep dive. It's not just about sweeping the streets. It's about fixing the city's crumbling infrastructure. When they popped the hood, they found that DDL357 had massively upregulated proteins vital for mitochondrial function.
Mitochondria, the powerhouse of the cell.
Exactly, the actual power grid of the city. In Alzheimer's, mitochondrial dysfunction is now recognized as a massive driver of cognitive decline. The neurons literally run out of energy and starve.
That's awful.
It is. But DDL357 significantly increased the expression of the literal biological turbines, specifically complex 1 and complex 3 of the respiratory chain that generate energy for the neuron.
Complex I and complex 3. So by upregulating them, the drug is essentially restoring the city's power grid.
Precisely. And they actually proved this in a separate test where they put human cells into an assay that measures oxygen consumption.
How did they do?
The cells treated with DDL357 showed a massive increase in their respiration rate. They were literally breathing better and pumping out more energy.
That is incredible.
But wait, there's more. If we connect this to the bigger picture, the proteomics also revealed an increase in 2 incredibly potent cellular VIPs.
VIPs, like protective proteins.
Yeah. For a 2-in-1 or SIRT1 and humanin.
SIRT1 and humanin. Let's break those down.
SIRT1 is a master metabolic regulator that coordinates overall cellular health and longevity. And humanin is a mitochondrial-derived peptide known for intense neuroprotective effects.
So enhancing both of these basically provides a robust, heavily armed defense against the cellular damage seen in Alzheimer's.
Exactly. But there's one final finding in the paper that truly shows the regenerative power of this molecule.
Oh, the neurite outgrowth. This blew my mind when I read it.
It's amazing. They took human iPSC-derived neurons, basically stem cells grown into actual human neurons in a dish.
Right.
When they applied the exact unbound concentration of DDL357 that naturally reaches the brain, the neurons physically grew.
They grew?
Yes. They promoted neurite outgrowth. They extended longer and more complex neurites, which are the branches they use to connect and communicate with other neurons.
It's literally rewiring the city. It's building new roads.
Exactly. It's not just stopping the damage, it's repairing it.
Wow. Okay. So what does this all mean for you listening? Let's zoom out and synthesize this whole journey. We started with a simple genetic clue. People with a mutation in the clusterin gene are highly vulnerable to Alzheimer's.
Well,
the researchers set out to artificially boost that protective protein, sCLU. They screened thousands of chemicals on human glioblastoma cells, found an epigenetic highlighter that hacks the cell's instructions, a BET inhibitor, and then spent months painstakingly engineering that chemical to survive the ADMET gauntlet and sneak past the fortress walls of the blood-brain barrier.
beautifully summarized.
And finally, they proved in living creatures that this single oral pill doesn't just clear the toxic trash. It heals the dying power grid by boosting mitochondria, protects the cells with SIRT1 and humanin, grows new neural connections, and genuinely restores spatial memory.
This research really represents a profound paradigm shift. As we mentioned, the current generation of Alzheimer's therapies are single-target snipers. They hunt down amyloid-β, and that is pretty much all they do.
Right.
But DDL357 is something entirely new. It is a multifunctional network drug. It modulates the entire proteostasis network, which is the body's master system for maintaining protein health, clearing out toxic aggregates, and balancing cellular energy.
It is a systemic fix for a systemic collapse.
And this raises an important question, perhaps the most exciting one of all, for the future of medicine.
Oh, I like where this is going.
Many of the most devastating neurodegenerative diseases we face, like Parkinson's disease, marked by clumps of alpha-synuclein, or ALS, marked by clumps of TDP-43, are fundamentally diseases of broken proteostasis.
Right, they all feature that toxic clumping.
Exactly. They're all caused by toxic cellular trash building up because the city's natural sanitation and power grids failed.
So I want to leave you, the listener, with this final provocative thought to ponder. We started this deep dive looking at the murky, tragic reality of Alzheimer's disease. But if a highly engineered, brain-penetrating molecule like DDL357 can successfully reboot the entire cellular infrastructure of a dying brain,
Could this exact same approach, maybe even this exact same molecule, one day be the master key to stopping Parkinson's, ALS, and the rest of these devastating protein clumping diseases?
It hints at a future where we don't just slow the dimming of the lights, but we actually turn them back on. Thank you for taking this deep dive with us.